Hey guys, welcome to AP Biology or College Biology. In this video, we're focusing in on Unit 5, Heredity, Section 5.3, which is all about Mendelian genetics. But before we get started, please note the disclaimer listed below. And also, if you're looking for reviews for earlier or later sections, please make sure to check out our podcasts and playlist section where we have organized videos for you. Also, make sure to hit that subscribe button for later videos just like this one. Okay, so let's get started. So let's talk about Gregor Mendel himself. Now, you don't need to know Gregor Mendel himself, but it's helpful to understand who he is and what he's about. So check it out. So he was a monk that basically used to farm. You don't need to know this information, but it helps paint a picture for you. So basically, he had a lot of time on his hands and he needed to make sure that he's able to understand as he was trying to conduct early science experiments to understand how genes pass on from parent to child. So he utilized pea plants. And the reason why he utilized pea plants is because there were a ton of different characteristics, as you can see here, like flower color and position and shape that were unique to pea plants, but a lot. They were varied. So, for example, you could have a tall pea plant or you could have a short pea plant. You could have a pea plant that is yellow or green in terms of their seed color. So these are all characteristics that you could see with the naked eye. And so it allowed Gregor Mendel to really study those pea plants easily. And also he could control mating because he could control the mating between the male and the female organs of the pea plants. So that was one of the benefits as well. Also, you could produce a lot of offspring over a period of time. It's not like humans where you have to wait a whole lifespan for something to grow and then continue to reproduce. Pea plants are pretty quick to produce offspring, which means that you could study them very easily. So, when we're talking about these pea plants, I just want to re-remind you of Unit 1, and this will come up in later units as well. But I just want to preface this by saying, remember the difference between DNA and RNA. Of course, they have nucleotides and they carry genetic material. But you need to remember that DNA ends up becoming RNA, which ends up producing proteins. So DNA contains our genetic material. That genetic material is then turned into RNA, which then turns into proteins, which is something that you need to understand when we understand what we're talking about with genetics itself. So let's go back to Gregor Mendel and his pea plant experiments. So what he did was he was trying to true breed, which means that when he was pollinating these plants, he was looking at the parent plants that have identical traits. So, for example, if one pea plant was tall, he would mate them with another pea plant that also was tall. If a pea plant had seeds that were wrinkled and other seeds that were not wrinkled, he would mate them together. And Gregor Mendel proposed several theories for what might potentially occur. And he had these, not theories, but hypotheses as to what would potentially happen with the pea plants. So the first one is called the blended hypothesis. And he said that basically the parent plants will merge together to create sort of like a hybrid with offspring. So you want to think of it as if we have a tall pea plant mixing with a short pea plant, then somewhere in the middle they would grow. Another one would be, say, for example, if we have two different colors, they would sort of blend together to produce a whole new color. So that's why there was an analogy of like paint per se. And that's his blending hypothesis, that the parental genetic material will merge together, both father and mother, and produce this genetic variation that would be a blend of both material. And the second theory that he had was the particulate hypothesis. And basically, what he said in that hypothesis is that parents will transmit very unique heritable traits that maintain their distinctiveness, which was the foundation of Mendel's theory. So basically, if you have a tall, that's unique for that tall plant. And if you have a short plant, it's unique for that short plant. If they blend together, they make an offspring that is unique of its own genes, and those passed through from parent to child. So it has a sense of distinctiveness, and that was the foundation of Mendel's theory. So he finally formulated what's called the law of segregation because he noticed that pea plants that had two various different traits produced genes or produced physical features that were characteristic of the dominant trait. So there is this ratio of 3 to 1. And we'll explain dominant and recessive in just a second and go into law of segregation. But it was the identification or understanding that law of segregation meant that both parents had their unique set of genes, and they would pass those genes depending on whether or not the specific gene was dominant or recessive. And that led to what we now know as the law of segregation. We did cover this when talking about meiosis. So if you remember in meiosis 1, in meiosis 2, it specifically when we're talking about chromosomes, we have chromosomes that are pairs, correct? And then what ends up happening is those pairs end up separating. And when they separate, they end up dividing as well. So you end up having 4 daughter cells during the process of meiosis that also because of independent assortment, but also due to law of segregation, you have 4 unique gamete combinations. And so that's what we now accept as law of segregation. So Mendel didn't have all the knowledge of meiosis and mitosis, but he had a theory in which he said his law of segregation was that the offspring had unique genes to the parents, but were from the parents in some way. Now, remember, we talked about diploids. Now, diploids are like us. We are 2N. We are considered diploid. And because we have 2N, we basically state that we have 2 alleles, 2 types of alleles, one from each parent that has been passed on to us. And then we will pass on a set of alleles onto our offspring, essentially. So now let's talk about the law of independent assortment. Remember, we talked about meiosis 1 and meiosis 2. So I'm going to go back to this slide here and we're going to focus in on meiosis 1. If you remember in prophase 1, what ends up happening is during the step of mitosis 1, we have prophase 1 in which we have the chromosomes swapping, right? So we have the pair of chromosomes, one from each parent. And if you remember, we could have, for example, these 2 homologous chromosomes. And maybe we have a little part that is swapped between both the ends. So that creates something unique as well. Well, remember during meiosis 1, we have those chromosomes ending up separating. And then in meiosis 2, we have the chromosome itself individually separating to create those 4 daughter cells. Okay, so what happens is the alleles of those genes are segregated independently during this gamete formation. So the four daughter cells, due to the swapping, creates and the idea that we're separating the segregation, we have gamete formation that is very unique. So the inheritance of one trait, such as flower color, also is noted to not affect another trait as well. So I want to point that out as well. So, for example, in some cases for flower color, say it's pink, and we know the flower is pink. We know that in some cases, due to the law of independent assortment, pink color does not affect something like pea plant color or pea plant shape, even though they're from the same plant. So, law of independent assortment says we have these genes, and oftentimes, this is not always the case, but oftentimes they, are going to be unique and unrelated to one another. So the principle of this is relevant to that genes are found on different chromosomes and they're positioned far apart from each other, so they're not going to have any relationship with one another. That's where that theory came from. But we do know that during meiosis 1 and meiosis 2, they end up separating, which creates these unique gametes, I should say. So now let's shift to the term genotype versus phenotype. Now, when we talk about genotype, we're talking about the genetic makeup of an organism. And the way that I like to think of it is genotype. We also have something called phenotype. Phenotype is the physical characteristics, something you could see with the naked eye. These are physical traits that can be seen, and that is noted as phenotype. So genotype is the genetic makeup. We can't see that with the naked eye per se, and then phenotype, which is the physical makeup. Okay, so now let's go over the different types of genotypes you could see. So we have two alleles. So they could either be two capital letters, two small letters, or a combination of a big letter and a small letter. Each one of these letters represents one from one parent and another one from another parent. So one from father, one from mother, to give you a unique combination of a genotype. So for example, let's say that you have big B, little b, or sorry, big B, big B. We know that the big B is representative of a dominant gene. So that's something else you need to remember. If it's a big letter, that means it's a dominant allele, not a dominant gene, a dominant allele. So when we have a small lowercase letter like little b, that indicates that it is recessive and it is not going to really show itself unless it's only the little letter case letters, essentially. So let me recap. So if you have two big B's, that means you have two dominants. So it's going to take on the personality of the dominants. So two big B's are also referred to as homozygous dominant. So say, for example, homozygous dominant means the color purple. Okay, so then it shows the color purple. If we know that little b means white and big B means purple, and they were to combine together, they would be called heterozygous. One allele from one parent and another allele from another parent to then give this unique combination of big B, little b. Once again, the big B means that it is dominant, so it takes on the characteristics of that big B, meaning purple. And therefore, phenotypically speaking, it once again will represent as purple. Now, if you have two lower letters, little b, that is considered homozygous recessive. Because we don't have the big B blocking the little b, you want to think of it as a bully. The big B is a bully, and covers up the little bee. But when we don't have bullies around, we just have two little bees. And that is called homozygous recessive, and those characteristics shine through. In this case, the flower is then white because little b represents white. So to recap, if we have the dominant feature, such as big B, regardless if it's heterozygous or homozygous, phenotypically, that big B will end up showing. So for this case, it shows up purple. If we have two little b's, the homozygous recessive traits end up coming through because they're not covered up by the big B in any way, shape, or form, and therefore defined as white. So this just shows how DNA influences our genes, which are representative of alleles, which show up on chromosomes. So that's the difference between genotype and phenotype and alleles. So when we talk about the law of segregation in humans, what he discovered when we talk about we talk about Gregor Mendel. Remember, he did true breeding. And he found some characteristics that are representative of our genetics. So if we have it's called an F1 generation. So that means from parent to offspring. This is the first generation. When there's true breeding, they'll notice that there's unique characteristics. And then F2 is from the second generation, or I should say the first generation to the second generation. That's called F2. He discovered that certain recessive traits would show up in those offspring. And so that's how he led to the formulation of law of segregation, which you'll cover up, or you'll see once you do things called Punnett squares. The law of segregation basically says that when you have the dominant and recessive, the dominant basically takes over. But we know that during reproduction, each parent will pass on one allele to its offspring. And then the combination of that dominant and recessive, or recessive or recessive, will basically produce and determine the phenotype of that offspring. So overall, the law of segregation ensures that gene copies are separate and allows for each gamete to only receive one specific allele from one specific parent, or each specific parent. So our parents basically provide to us our genes, and they provide to us a copy of one of their genes, and those genes are then passed on to us. So when we begin to review that, we could see that there are certain things that we need to remember in terms of the relationship between DNA all the way down to recessive allele. This copy of our chart here really shows the relationship between what we talked about, that DNA is in our chromosomes. Chromosomes formulate our genes. The genes have certain alleles that could be passed on. They create certain genotypes, which are genetic background. Those combination of certain alleles that formulate the genotype can eventually distinguish themselves in physical expression called phenotype. And the phenotype is dependent on the dominant allele versus the recessive allele relationship. So I hope that helps really clear up some terms there. Now let's try to complete a quiz question together. Before we get started, I want you to understand the two following terms you see here. The first one is called monohybrid, and the second term is called it's called a monohybrid cross, and the second term I want you to know is a dihybrid cross. A monohybrid cross is going to be pretty easy to understand. It's usually one allele with each other. Now, if you remember, we talked about the idea that one gene does not affect another due to the law of segregation. However, there are certain traits that can be related to each other, and they're called dihybrid crosses. For example, if we said that a flower color like purple was related to the shape, such as round, we would mix the two together to create a unique combination of alleles from both parents, such as heterozygous from one parent, and heterozygous for another parent. Okay, let's try to attempt a Punnett square question. Take a moment, pause this video. I know we haven't done any questions related to Punnett squares just yet, but I want you to take a moment and try to read the following question and see if you can answer it yourself. Once you're done, unpause the video and come back here. Okay, so let's tackle the following question, which basically is a type of monohybrid cross. So in this question, it states in a breed of cats, a gene, big B, determines fur color. The dominant allele, big B, codes for black fur, while the recessive allele codes for white fur. Suppose a heterozygous black furred cat mates with a homozygous recessive white furred cat. So they're talking about fur. It's one characteristic. So this is going to be a monohybrid cross. They tell what the parents are, which are heterozygous recessive white furred cat and homozygous recessive white furred cat with a heterozygous black furred cat. So first I'm going to tackle the heterozygous black furred. So remember, black is considered a dominant gene. So we have big B, and I should make my big B a little bit better than that. So we have big B, and it's heterozygous, which means the opposite of homozygous hetero means it's two different. So it's big B, little b. And it's going to combine with a homozygous recessive, which lets us know that it's little b, little b. What you're going to do is we have to figure out the genotype ratio and the phenotype ratio in this question. So we're going to do a cross. The way that you set up the cross is, I'm going to use a different color here. We have to go ahead and create our Punnett squared as so. And we're going to, probably I should make this a little bit bigger so you can see a little bit nicer. So we're going to make, usually I make a box and I draw a little crisscross just like this. Now, the one parent is going to have big B, little b, and the other parent, if you remember, is little b, little b. So we have both our parents set up. It could be the horizontal or vertical. It doesn't matter as long as one parent is on the one side and the other parent is on the other side. Now we have to go ahead and combine both of them. So we have in this first box, big B and little b. And then we bring down the big B here with the little b here, and then little b here with little b, and then little b here with this little b. So you could see here, we take one from each parent to match up each one of these boxes or cells. So this allows us to give us our genotype, which is 2 big B, little b. And the reason how I found out 2 big B, little b is because I just counted up the big B, little b. And then we do a little quote or little punctuation, like 2 dots like that, with little b, but then how many do we have? We have 2. So we're going to go say it's 2 little b, little b. So for every 2 big B, little b, there's 2 little b, little b. Now you could reduce this because it's a 1-to-1 ratio. And that's what we're doing here. So genotypically, you could say 1 big B, little b for every 1 little b, little b. And so that is the genetic ratio. Now, phenotypically speaking, we have to now analyze this to say the dominant gene is the big B. So we know that for every 1 black fur, we have 1 white fur. Okay, so that's 1 black for every 1 white. So you could see here we have 2 big B, little b to 2 little b, but it's also the same way as saying 1-to-1. And I just want to point that out as well, because you could get a multiple choice question and you need to understand that these can be reduced to 1-to-1, or it could be same thing with 2-to-2. They're both the same value. Okay, so let's try to conduct a dihybrid cross. This is when you have two mice with two characteristics in this case that need to cross with each other. So take a moment, pause this video, and unpause once completed so we could tackle this question together. Okay, now that you're done, let's review the question together. In this question, it states in mice, black fur, big B, is dominant to brown fur, little b. In short tails, big B is dominant to long tails, little t. And if we cross the mice, what fraction of the offspring will have big fur and long fur? So I went ahead and set up the cross so you could see the dihybrid cross here and how it looks and your potential answer for what the fractions are. And I want you to understand that we have these two parents here. And to set up your dihybrid cross, you need to understand it's a 4 by 4. So you're going to have 1, 2, 3, 4 by 4. And you're probably wondering how we got these parents listed. So you want to think to yourself that you're taking big B with little b with big T with little t. So we have to tackle this first one because they're both the same, big B, little b with big T, little t. I can basically set up one parent and know that the same is going to be for the other parent, right? Now, this will not be always the case, but in this case you can. So you have big B with big T because you do the first with the first, so big B, big T. First with the second, so that's big B, little t. So I use the foiling method. So first, outer, inner, last, or first, inner, outer, last. You can remember it as FOIL, but just to recap, I'm taking all possible combinations there are, and I'm going to create that dihybrid cross by taking the first with the first, big B, big T, first with the second, so big B, little t, little with the inner with the first, so that's little b, big T, and last with the last, so little b, little t. You could think of it as first, the outers, the inners, and the last, the big B and the little t. So either way, you could use that method of FOIL to help you to remember the combination of orders, but you're just combining them all, and you're going to set up a dihybrid cross. And then once you do, you're collecting all of the combinations like this one here, and you're basically looking for which ones will have black fur and long tails. Black fur is going to be your dominant, right? So you don't want any sort of recessive to little b's in here to count. So I usually just eliminate those off the bat and I can get rid of them. So that's one way to get rid of them. And it says that has long tails, which are just recessive. So that means little t, little t. So we can eliminate anything that has big T. So I can eliminate that one, 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 eliminate that one. And as you can see here, we are now left with 3. 1, 2, 3 out of the total number of boxes, which is 16. So you get 3 over 16. I did set up from fractions here where you can also do it by setting up 2 monohybrid crosses. That's one way to do it. You could do big B, little b times the other big B, little b, and then set it up where you're just taking a look at the T's, cross them together, and multiply the percentages that you get for each of the characteristics. That is an alternative way of solving as well if you choose to do it that way as well. So I hope this helps understand the differences in monohybrid versus dihybrid crosses and Mendelian genetics. Hopefully you'll stick around for later videos to understand the later units, and I hope this helps. Thank you. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams.